Hello everyone, my name is Matthew Berardi, and I'm going to be talking to you about the medical microbiology of the current COVID-19 pandemic. Before we delve into the underlying biology, it's necessary to clarify a few important terms. Contrary to their interchangeable use in everyday vernacular, COVID-19, SARS-CoV-2, and coronavirus each have specific and non-overlapping scientific meanings. COVID-19, for instance, is the name of the disease that has spread across the world over the past few months, while SARS-CoV-2 is the virus that causes this illness. It is a novel coronavirus, a term that describes a group of viruses so named because of the resemblance to a solar corona. Now that we understand the terminology, let's take a more detailed look at coronaviruses. Coronaviruses belong to the Coronaviridae family of the Nidovirales order. The Coronavirinae subfamily with which we are presently interested is further divided into alpha, beta, gamma, and delta classifications. On the left, we see examples of the positive sense RNA genomes that all coronaviruses possess. In addition to serving as the heritable genetic material, the RNA can be directly transcribed by host cell ribosomes. There are four major structural proteins for which it codes, including the characteristic spike protein. This protruding structure on the viral surface contains a receptor binding domain that interacts with host cells. Membrane proteins are the most abundant. These transmembrane dimers are responsible for the shape and assembly of mature particles. Envelope proteins, meanwhile, are involved in viral assembly and release, while nucleocapsid proteins package the genome and tether it to the replicase-transcriptase complex, which we will explore in more depth later. The coronavirus infection cycle begins when spike proteins bind to host cell receptors. A protease then cleaves the S protein, allowing the viral membrane to fuse with either the host cell's plasma or endosomal membrane. The viral genome is released into the cytosol, and the replicase gene is first transcribed by the host ribosomes. The replicase-transcriptase complex then assembles, enabling the synthesis of both genomic RNA for later packaging and subgenomic RNAs that encode structural and accessory proteins. At this stage, recombination of identical RNA or genetic material from different viruses infecting the same host cell is common, a process which is believed to promote viral evolution. Next, newly translated S, E, and M proteins are inserted into the endoplasmic reticulum membrane. Mature particles are then formed when N proteins and associated RNA genomes enter this compartment. Golgi vesicles transport them to the cell surface, where exocytosis allows their release. This cycle causes a variety of diseases in mammals. Piglets, for instance, are susceptible to both transmissible gastroenteritis virus and porcine epidemic diarrhea virus, while porcine hemagglutinating encephalomyelitis virus can infect the swine nervous system. The respiratory disease and diarrhea associated with bovine CoV has devastated the cattle industry, while murine hepatitis virus has been utilized by researchers to model several human ailments, including respiratory disease and multiple sclerosis. Scientists even discovered a novel coronavirus known as SWI by sequencing the diseased liver tissue of a dead beluga whale. As seen in the figure on the right, humans have been infected with several animal coronaviruses. HCoV-229E, NL63, OC43, and HKU1, for instance, are responsible for around 1/4 of annual respiratory tract infections. Recently, however, there have been several deadly outbreaks of new coronaviruses. SARS-CoV, the causative agent of severe acute respiratory syndrome, or SARS, is understood to have originated in bats and spread to humans through civets. Researchers believe that MERS-CoV, the virus that produces Middle East Respiratory Syndrome, or MERS, also originated in bats and was transmitted to humans through camels. As we'll discuss shortly, the origin of SARS-CoV-2 is uncertain, though it is closely related to a bat coronavirus. Let's now take a closer look at some of these severe illnesses. SARS and MERS, which are both characterized by fever, cough, and difficulty breathing, have emerged as highly feared diseases over the past 2 decades. SARS results from the binding of a beta coronavirus to ACE2, a receptor found on cells of the lungs, kidneys, and intestines. The contagion originated in the Guangdong province of China in 2002 and spread to 28 other countries over the next several months. Though no new cases have been reported in over 15 years, 9% of the 9,098 infected individuals died from SARS. The binding of another beta coronavirus to the DPP4 receptor of lung and kidney cells leads to MERS. This deadly illness propagated from Saudi Arabia to the surrounding region in 2012, and another large outbreak occurred in South Korea in 2015. With a mortality rate of a staggering 35%, this ongoing disease has killed over 800 people to date. SARS-CoV-2 is another beta coronavirus, representing a new strain from the same species as SARS-CoV. However, it's most similar to bat-CoV RATG13, a bat coronavirus with 96.2% of the same nucleotides. This remarkable correspondence has generated speculation that the virus began to spread in Wuhan, China after passaging to humans. A new non-peer-reviewed study indicates that over 30 strains of SARS-CoV-2 could be circulating, each with variable viral loads and pathogenicity. Researchers have determined that the virus binds to ACE2 with similar affinity as SARS-CoV, though the spike protein's novel furin cleavage site could account for the increased transmissibility. A defining feature of deadly infection is cytokine storm, which results from the release of these small signaling proteins by white blood cells. Their recruitment of more white blood cells to the upper respiratory tract causes cell death and inflammation of the lungs and heart, resulting in pneumonia or ARDS, which I'll discuss in the next slide. According to virologist Peter Kolchinsky, the virus's spread is enhanced by its initial asymptomatic infection of throat cells, leading to person-to-person transmission before symptomatic lung invasion. According to the Centers for Disease Control and Prevention, SARS-CoV-2 is primarily transmitted from person to person through respiratory droplets. However, contaminated surfaces may also present a secondary means by which the virus is spread. CDC reports that symptoms typically appear 4 to 5 days after infection. Common signs of COVID-19 include fever, cough, fatigue, and lack of appetite, while loss of taste or smell have also been noted. 19% of affected individuals are hospitalized, while 6% are admitted to the ICU. Those who develop difficulty breathing typically do so about a week after infection, while acute respiratory distress syndrome, or ARDS, generally manifests after 8 to 12 days. Preliminary data from the U.S. indicate that age is among the most important risk factors, with 10 to 27% of patients 85 and over dying due to COVID-19. That rate decreases to between 3 and 11% for those aged 60 to 84, while less than 1% of diseased individuals under 54 have died. Epidemiological data from China indicate that cardiovascular disease, diabetes, chronic respiratory disease, and hypertension are all associated with poorer outcomes. Fortunately, 81% of COVID-19 cases result in mild to moderate symptoms, while 14% are severe. Case fatality is nearly 50%, however, for the 5% of patients who experience critical illness. Asymptomatic transmission of the virus has received much recent attention, and an estimated 25% of affected individuals do not experience symptoms. Now let's take a look at the current status of the COVID-19 pandemic. The Center for Systems Science and Engineering at Johns Hopkins University reports that there are over 2.7 million confirmed cases and 195,000 fatalities in 185 countries and regions across the globe. In the United States, nearly 900,000 cases have already been confirmed, with over 30% concentrated in New York State. Sadly, 51,017 Americans have died from the virus. Countries around the world have restricted their populations to control the virus's spread. China suspended travel into and out of Wuhan in late January, while Italy implemented a national lockdown on March 9th. California issued the first stay-at-home order in the U.S. on March 19th, and 42 states eventually followed suit. Currently, an estimated one-third of the world's population is constrained in some manner. Face masks, long more common in Asian nations, are now recommended by CDC for use in public due to the prevalence of asymptomatic carriers. Typically using nasopharyngeal swabs to gauge the current presence of the viral genome, over 4.4 million RT-PCR tests have been performed in America. Large-scale serology tests, which detect antibody responses, have recently shown that as much as 5% of Los Angeles County and 21% of New York City have been exposed to SARS-CoV-2. This could indicate that the death rate is much lower than previously assumed, but the protective immunity of these antibodies is still unclear. Clinical trials to repurpose known pharmaceuticals, like the arthritis drug tocilizumab, are now underway. After initial success in small samples, the benefits of hydroxychloroquine and remdesivir now seem dubious. Whereas earlier models once predicted a much deadlier fate for the United States, the frequently referenced projection from the University of Washington's Institute for Health Metrics and Evaluation now foresees 67,641 COVID-19 deaths in the United States by August 4th if containment measures are continued. Fear persists about a second wave of infection this fall, however, and scientists are racing to develop antiviral medications. Researchers at Northwestern University, for instance, have proposed several SARS-CoV-2 protein targets, including an endonuclease, replicase, and methyltransferase, while a team in Austria is testing a recombinant version of the ACE2 receptor to block viral entry. Several vaccines are also in development, including ones that utilize mRNA and DNA plasmids, though they are unlikely to be approved for 12 to 18 months. Over the past few weeks, several European countries have begun to gradually ease their stringent lockdowns as the number of new daily cases has fallen. Smaller stores have already reopened in Germany, while 300,000 Spaniards resumed work last week. These changes are far from immediate returns to normalcy, though, as the partially reopened Shanghai Disneyland requires that visitors wear masks and undergo temperature screenings. The South Korean Baseball League may provide a model for the reintroduction of professional sports in the United States, with the preseason featuring empty stadiums and umpires donning masks and gloves already underway. Though the easement of similar measures across much of the U.S. is still unlikely for weeks or months, some non-essential businesses have already reopened in Georgia, Alaska, and Oklahoma. Here are the sources for this presentation. Thanks for watching.